Capillarity Lab. Drying soil samples. Many measurements of soil, including this one, require drying the soil prior to the measurement. Otherwise, water in the initial sample may affect your results. Drying can be done by either spreading the sample in sunlight for several hours, placing the sample in a 105-degree oven for several hours, or placing it in a crucible with a Bunsen burner until it's dried. Soil capillarity is the rise of water in a soil due to capillary action. Whereas porosity increases with grain size, soil capillarity decreases with grain size as capillary action is enhanced by small space between particles. We will be determining how to measure the comparative soil capillarity between 3 soil samples. 3 identical cylinders are prepared. They are open at both ends, and cotton wool is used to seal the bottom of each so water can enter the bottom of the tube, but the soil does not fall out. A different soil sample is added to each tube. A little soil is added each time, and the tube is lightly tapped to compact it well. Each tube is labeled so the results of the test can be related to the soil sample. Then, all 3 tubes are placed in a dry basin. In this case, they are labeled A, B, or C. Note that the tubes are all arranged to be the same height above the bottom of the basin, and they do not touch the bottom of the basin. This is accomplished by having each tube held by a ring stand, not shown. Water is then added to the basin and a timer is started. As water rises in the tubes due to capillary action, its progress can be seen by the soil changing color as it becomes wet. Periodic measurement will reveal how quickly the water rises in each tube. After enough time passes, hours or even days, the relative height of the column of water shows which soil has more capillarity. The higher the water rises, the more soil capillarity. In this case, the highest soil capillarity is found in sample C, and the least is found in sample A.